Welcome to Biologics. This is the second part of the tutorial Basics of Genetics. To learn about Mendelian genetics, don't forget to watch the first part. But before we begin, subscribe to the channel for more educational content every week. Let us begin. In this tutorial we are going to learn about chromosome theory of heredity, phenomenon of linkage, crossing over, and recombination, linkage maps, sex determination mechanisms, chromosomal disorders. Mendel's experiments made it clear that in heterozygous genotypes neither allele is altered by the presence of the other. Also, the hereditary units remain stable and unchanged in passing from one generation to the next. However, at the time the biological basis of the transmission of genes from one generation to the next was quite mysterious. Now a question arises: how did the chromosome theory take shape? To address that, evidence is accumulated from a variety of sources, and when microscopy had improved enough, the next major advance came with the discovery of chromosomes, and one of the first lines of evidence came from observations of how chromosomes behave during the division of a cell's nucleus. Credit for the chromosome theory of heredity, the concept that genes are parts of chromosomes, is usually given to Walter Sutton and Theodore Boveri. Working this time with meiosis, in 1902 these investigators recognized independently the behavior of Mendel's hypothetical particles during the production of gametes. In 1902 and 1903, Sutton and Boveri published independent papers and stated that individual genes are found at specific locations on a particular chromosome, and that the behavior of chromosomes during meiosis can explain why genes are inherited according to Mendel's laws. The first indisputable proof that genes are parts of chromosomes was obtained in experiments conducted by Thomas Hunt Morgan in 1910, for studying the pattern of transmission of the eye colors in Drosophila. The studied gene was proved to be present in the X chromosome, and a mutation affected fly eye color, resulting in formation of white eyes. He observed that the mutation was inherited differently by male and female flies, because it is the X chromosome that is transmitted in a different pattern by males and females. A male transmits his X chromosome only to his daughters, whereas a female transmits one of her X chromosomes to the offspring of both sexes. A similar example of a human X-linked trait is hemophilia, a severe disorder of blood clotting determined by a recessive allele. In the early 1900s, William Bateson and R.C. Punnett were studying inheritance of two genes in the sweet pea. In a standard self of a dihybrid F1, the F2 did not show the 9:3:3:1 ratio, predicted by the principle of independent assortment. In fact, Bateson and Punnett noted that certain combinations of alleles showed up more often than expected, almost as though they were physically attached in some way. They had no explanation for this discovery. Later, T.H. Morgan found a similar deviation from Mendel's second law while studying two autosomal genes in Drosophila. Morgan proposed a hypothesis to explain the phenomenon of apparent allele association and attributed this due to the physical association or linkage of the two genes, and coined the term linkage to describe this physical association of genes on a chromosome. The work of Morgan showed that linked genes in a dihybrid may be present in one of two basic conformations. In one, the two dominant or wild-type alleles are present on the same homologue. This arrangement is called a cis conformation. In the other, they are on different homologues, in what is called a trans conformation. To understand the phenomenon of linkage, let us study a gene that affected eye color and wing length. Here, PR codes for purple, and PR plus for red, while the wing length is represented by VG for vestigial wing, and VG plus for normal length. The wild-type alleles of both genes are dominant. Morgan suggested that the two genes in his analysis were located on the same pair of homologous chromosomes. Thus, in the first analysis, when PR and VG are introduced from one parent, they are physically located on the same chromosome, whereas PR plus and VG plus are on the homologous chromosome from the other parent. In the second analysis, one parental chromosome carries PR and VG, and the other carries PR plus and VG plus. The linkage hypothesis explains why allele combinations from the parental generations remain together, because they are physically attached by the segment of chromosome between them. But how do we explain the appearance of the minority class of non-parental combinations? Morgan suggested that when homologous chromosomes pair in meiosis, the chromosomes occasionally break and exchange parts in a process called crossing over. Morgan suggested for non-parental combinations, that when homologous chromosomes pair in meiosis, the chromosomes occasionally break and exchange parts in a process called crossing over. This physical exchange of chromosome segments results in formation of two new combinations, called crossover products. In 1931, Harriet Creighton and Barbara McClintock, while studying two genes of corn, located on chromosome 9, that affected seed color, C, colored, C, colorless, and the other affected endosperm composition, WX, waxy, WX, starchy. In the progeny of a test cross of this plant, they compared the chromosomes carrying new allele combinations with those carrying parental alleles. They found that all the recombinants inherited one or the other of the two following chromosomes, depending on their recombinant makeup. Thus a precise correlation between the genetic event of the appearance of new allele combinations and the chromosomal event of crossing over. The chiasmata appeared to be the sites of exchange, although the final proof did not come until 1978. Here we can see the diagrammatic representation of crossing over, showing a crossover taking place at the 4-chromatid stage of meiosis. However, it is theoretically possible that crossing over occurred at the 2-chromosome stage, before DNA replication. This conundrum was resolved after results of tetrad analysis, which indicates that crossovers occurred at the 4-chromatid stage because if crossovers occurred at the 2-chromosome stage, there could only ever be a maximum of 2 different genotypes in an individual tetrad. The occurrence of more than 2 different products of single meiosis seen here in tetrad analysis indicated that the crossing over occurred only after the replication of DNA. The term recombination is used to describe the generation of non-parental gene combinations. Recombinants are produced by 2 different cellular processes, independent assortment and crossing over during meiosis. Meiotic recombination is defined as any meiotic process that generates a haploid product with new combinations of the alleles carried by the haploid genotypes that united to form the dihybrid meiocyte. Thus, any meiotic product that has a new combination of the alleles provided by the 2 input genotypes is by definition a recombinant. If we take a look at the comparison of 2 meiotic products, one where crossing over did not occur and the other where crossing over occurred, we would observe that without crossing over the parental combination of alleles was retained, while with crossing over, 2 new reciprocal recombinants were obtained in addition to the parental combinations. To understand how recombinants are produced, let's reconstruct a standard Mendelian analysis. A recombinant frequency of 50% in a test cross suggests that the 2 genes under study assort independently. The simplest and most likely interpretation of independent assortment is that the 2 genes are on separate chromosome pairs. In Morgan's study, the linked genes could not assort independently, but there were still some recombinants, which as we have seen, must have been produced by crossovers. As seen in the second figure that a single crossover generates 2 reciprocal recombinant products, which explains why the reciprocal recombinant classes are approximately equal in frequency. Thus, we may comment that a recombinant frequency significantly less than 50% indicates that the genes are linked, while a recombinant frequency of 50% generally means that the genes are unlinked and reside on separate chromosomes. Linkage maps. Morgan studied more linked genes, and he saw that the proportion of recombinant progeny varied considerably depending on which linked genes were being studied. Morgan thought that such variation in recombinant frequency might somehow indicate the actual distances separating genes on the chromosomes. He assigned the study of this problem to a student, Alfred Sturtevant. Sturtevant postulated a rough proportionality: the greater the distance between the linked genes, the greater the chance of crossovers in the region between the genes and, hence, the greater the proportion of recombinants that would be produced. Thus, by determining the frequency of recombinants, we can obtain a measure of the map distance between the genes. Sturtevant define one genetic map unit, mu, as that distance between genes for which one product of meiosis in 100 is recombinant. Put another way, a recombinant frequency, RF, of 0.011% is defined as 1 meter.u. A map unit is sometimes referred to as a centimorgan, cm, in honor of Thomas Hunt Morgan. Now, let us apply Sturtevant's postulate taking the case of three genes, A, B and C. To know their arrangement on the chromosome, let us use the estimated recombination frequency, which is observed to be 5 map units, separating genes A and B, whereas 3 map units separate genes A and C, then B and C should be either 8 or 2 map unit apart. Sturtevant's analysis strongly suggested that genes are arranged in some linear order. The place on the map, and on the chromosome, where a gene is located is called the gene locus, plural, loci. Sex determination mechanisms. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. Hinking, in 1891, traced a specific nuclear structure all through spermatogenesis in a few insects, and observed that only 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hinking named this structure as the X-body, but he could not explain its significance. Later, cytological investigations for X-body revealed that it was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that the ovum bears 2 copies of the X chromosome while the sperm bears only 1. Ovum fertilized by sperm having an X chromosome become a zygote that grows up to become female, while those fertilized by sperms without X chromosome become males. Considering the role played by X chromosome in sex determination of the zygote, it was designated as sex chromosome, and the rest of the chromosomes were named as autosomes. Later studies in humans and Drosophila revealed the presence of a homologue of X chromosome, which was named as Y chromosome and was present in sperms without X chromosome. Investigations in a number of other insects such as Drosophila and mammals including man, revealed the presence of XY type of sex determination mechanism in these organisms. Humans and Drosophila have a smaller homologous pair for X chromosome, called Y chromosome. Both male and female have the same number of autosomes. However, human males have 1 X and 1 Y chromosome, while the female has 2 X chromosomes. Human males exhibit heterogamity and thus, during the mitogenesis produce sperms bearing 1:1 ratio of X and Y chromosome bearing gametes. Thus the chance of the fetus being born male is 50%, and is a random occurrence. Chromosomal disorders. The chromosomal disorders are caused due to absence, or excess or abnormal arrangement of one or more chromosomes, which might occur due to the failure of segregation of chromatids during cell division. The failure in segregation often results in the gain or loss of a chromosomes. This gain or loss results in aneuploidy. Failure of cytokinesis after the telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. Sometimes, although rarely, either an additional copy of a chromosome may be included in an individual or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome. Trisomy or monosomy of a chromosome often has serious consequences on the individual suffering from them. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, trisomy of 21. In the karyotype shown here we can see that the individual has 3 copies of the chromosome number 21. This disorder was first described by Langdon Down, in 1866. The affected individual is short-statured with a small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Klinefelter's syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting in a karyotype of 47, as visible in the karyotype that the individual has a sex chromosome combination of X, Y, Such an individual has overall masculine development. However, the feminine development, that is, gynecomastia, is also expressed. Such individuals are sterile. Turner's syndrome. Such a disorder is caused due to the absence of one of the X chromosome in females. Consequently, the individual has a total of 45 chromosomes instead of 46. Such individuals are represented with exogenotype. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters. Thank you for watching. I hope you have enjoyed learning the basics of genetics. Like, share and subscribe Biologics for more e-learning contents. See you next time.